I think I will just say one or two sentences so that I leave as much time as possible to Cori, but that is where it's very fashionable to discuss about big ideas and simulate huge networks and so on. It's really very, very inspiring to see somebody who can extract such deep statements, apparently simple organism. And Cori has done a huge amount of work that is covers a huge amount of different scientific levels, epistemic levels, and on so-called simple organisms, going from the molecular to the behavioral. And I will just give as an example that I was teaching in a summer school a couple years ago with many economists and cognitive scientists. And I wanted to show some example of when internal states and internal variability matters for producing behavior. And looking in the literature, I ended up on some work of Cori, which I did present in these lectures. And I had some questions, and I had never met Cori, and sent Cori some questions by email and got very rapidly, very detailed and very kind responses. And so I noticed that she's not only a great scientist, but also a wonderful human being, and very kind and very attentive. So with this, I think we should let you speak, Cori. Thank you very much.
Thank you. Thank you for that very kind introduction, Rava. Yeah, he reminded me of that correspondence when I was invited to this course. So my career started as a graduate student working in cancer biology with Bob Weinberg, and then I switched to working on behavior as a postdoc with Bob Horvitz at MIT. And I was completely drawn into the system of the nematode worm Caenorhabditis elegans and the idea of using that to understand the nervous system and behavior, and that's where I've been ever since. I'm excited to be at this course because I think a lot at the moment, at this point in my career, about what the future of the field is like, what the next steps are, and what we want to do as a community. And I really believe that theoretical and computational approaches are going to be incredibly important tools for the next stage of biology. And the more people can master them, the more insights they're going to be able to get for understanding not just the different phenomena that go on, but the principles that underlie them and sort of some of the hidden rules of biology that we can't yet name and describe. So I think this is a great course. I'm really glad that this is sort of putting together biologists and experimentalists. I am very, very much an experimental biologist myself. And so the topic of my lecture today is going to be a series of observations about the nematode worm C. elegans and about its behavior and about its nervous system and the molecules and the properties of the neurons, and that I will pose as questions that have sort of initial level descriptions that are qualitative and not terribly precise, and will really benefit from a deeper description that comes out of the kinds of approaches that you all are addressing and learning in this course. So I will share my screen. And move to the first slide. Is everything on?
Yes.
Yes, thank you, Trent. And and can you see the cursor?
Yes.
Yes, we see everything.
Okay, thank you. So here is the logical framework that I'm going to be using to describe the work that my lab does and the work that I'm going to tell you about today. And I'm going to describe a very general problem, which is that behavior emerges on many different biological scales. So if we think about what the nervous system is doing, it is at times operating at a time scale of channels and synapses that carry out their functions in fractions of a second. And then at other levels, the nervous system, like all organs in the body, is defined by the genome and by evolution, which takes place over millennia and millions of years to shape the properties of the nervous system and to guide it toward adaptive behaviors. So in between here are many of the things that, for example, a psychologist might think of as a behavior. So for example, perception, computation, and action. These are things that happen with amazing speed. If the telephone rings and you pick it up and you hear your mother's voice, you recognize that, in under a second, you have the processing of the information, who it is, the timbre of the voice, and place that association with the person that you have a memory of. It's amazing how fast these processes happen. So there's this rapid flow, but there's also interesting slower flows of information through the nervous system. For example, all of these kinds of behaviors are shaped, as Rava very kindly mentioned, by processes like emotion and motivation and behavior that change the outcome of behaviors, not in that sub-second time scale, but over longer and more stable time scales. And at still longer time scales, you have phenomena like learning and memory and development that reshape the nervous system to sort of send it toward different directions and give it different biases in the future. Now, in the areas of computation, I would say that this question of timescales and time series is probably one of the ones that is still least well described and understood at the moment. And I think it's an important area for us to understand because timescales are important in all of biology. And determining how these different systems interact, how they talk to each other, and different phenomena that arise at these different levels and how is what's going to be the topic of the talk. Now, I'm a neuroscientist, and as a neuroscientist, I'm a human neuroscientist, so I'm really interested in the human brain and human behavior. But I'm daunted by the level of analysis that you're going to be able to make by studying the human brain. It's extremely complicated, it's extremely large with billions of neurons and trillions of connections. And so what drew me to C. elegans as a postdoc and has kept me there is the fact that its nervous system is completely described, is all available to us anatomically. And this illustration here actually is a description of work that was published right when I started my postdoc, which was the wiring diagram of C. elegans. So each of the 302 neurons in the animal's nervous system was followed through serial section electron micrographs, and all of the connections between those neurons were determined, and sort of a set of logical relationships and anatomical relationships were described. And here it was, a complete map of an animal's nervous system at an anatomical level that described many of the ways that information could flow through it. And it gave me a sense of what the kinds of problems were that we could solve. And I think it still serves as an incredible framework for this solution. So what is it you're looking at here? You're looking at a diagram of the nervous system with some of the different neurons color-coded. So for example, about a third of the neurons are sensory neurons. They sense different chemical stimuli, mechanical stimuli, light, a few other kinds of inputs. And this is kind of the standard drawing of a nervous system, which goes from sensory at the top down to motor at the bottom, because about a third of the neurons shown in green are motor neurons. And these are the different kinds of systems that move the worm around. And to a first approximation, when we study worm behavior, we actually track a set of very simple motor actions, although there are more complex ones. We figure out whether it's moving forward, whether it's moving backward, or whether it's making a turn, how often those things occur, and how quickly individual actions are carried out. And then in the middle, in blue, are a set of integrating neurons. They're the ones that process that sensory information and lead it to the motor output. And a lot of what we're really interested in and a lot of what's hard to solve are the steps that occur at these integrating neurons. That's where the big challenges lie. Just to mention that there's also a little part of the nervous system involved in feeding in the pharynx, off there to the left, about 30 neurons that are only connected by a single synapse to the rest of the nervous system. So the thing that's important about this wiring diagram is that everyone agrees on what it is. Everyone agrees on what these neurons are. And these neurons are distinguished from each other in ways that, you can go in to look at a particular neuron. I'm going to pick one out right here in red, the AWA sensory neuron. And when I say what that neuron is, I know what its position is. I know what genes it expresses. It's going to express those genes reliably in every animal. And I know what functions it has and what behaviors it's involved in carrying out. And so that is illustrated here. Here's the AWA neuron. It's in the head of this worm here, along with most of the neurons in the worm's nervous system. It has an ending that goes to the tip of the nose, and it is activated by certain odors that C. elegans finds extremely attractive, including the odor of butter, diacetyl, and C. elegans will chemotax toward those odors in a behavior that is completely dependent on this AWA sensory neuron. And then right next to it is another neuron that detects noxious substances. Those noxious substances are avoided by the worm. And then underneath that, right next to that is a neuron in blue. That's another neuron involved in detecting attractive substances, AWC. AWC expresses a different set of genes and detects a different set of odors. So it mediates chemotaxis to a different pool of the odors in the environment. It actually has a different signaling component, cascade. It's inhibited by odor instead of activated, and it also mediates chemotaxis. So we can start with this kind of information about sensory neurons, and don't worry about the names particularly. I'll try to remind you each time I talk about something, like I'm talking about an olfactory neuron now. But what I'm really trying to explain is something like the track at the bottom of this image. And this is the track of an animal that was put here. And you came back about 15 minutes later, and the animal had reached the peak of a gradient of odor. And it had done that with accuracy, even though this worm is a millimeter long, and this path here is probably about 100 times as long as the animal was. And you can tell that it wasn't a completely straight path. It was sort of a complex path. There are different components of these forward and backward and turning behaviors that somehow got it to what it was. And at some level, just explaining that was where we wanted to start. Now, the thing about this is that this is the way we used to study C. elegans behavior, in this kind of qualitative way. You put an odor down on a plate, you knew how much you put down, you didn't know exactly how it was diffusing through the system, but the animal responded to it. And sort of the next level of understanding of behavior, where we are now in C. elegans in all animals, is trying to rigorously control the stimuli that we give to the animal so that we can really understand how those stimuli are processed. And so this was a project that was, pardon?
May I quickly ask you a question?
Sure.
And
yes.
also, I should have asked you before, do you prefer if students interrupt you with questions, or do you prefer them to write in the chat and then we moderate? Is it okay if we just interrupt you?
It's fine to be interrupted. It's not, if you write in the chat, I need you or Twen or someone to alert, and then
Sure.
maybe have the student say the question out loud, because I can't really run the chat and the
No,
slides
that's
and
what
look
I
at
meant.
pictures at the same time. Yeah.
Of course, that's what I meant. So either please interrupt, or if you write in the chat, I'll interrupt. Just one quick question about the diagram. One is always presented with this diagram, but is there must be some variability from individual to individual. Can you just say a word about that so that I have an idea of how that?
Yeah. So yeah, no, that's a great point, Rava. So there's this wiring diagram, and it was like, the resource that everyone went to. It was the ground truth of the wiring diagram, and it was based on basically parts of 3 worms, and usually no more than 2 worms at any one location. And even within those, there was a certain amount of variability between the different connections. And recently, this has really been scaled up a lot. People have done a few more worms. There are just 2 papers that appeared that started to look at worms across development and look at some of these other connections. And the simple conclusion is that the general patterns of connections and proximity are the same between different animals, but they're not identical. So if you count synapse for synapse, there are about 70% of the ones that are found in one individual are found in all of the others. But for example, that's just at the level of the connection of one neuron to another. If you count the number of those connections, one animal might have 3 connections and the next one might have 9. Okay, 9 between a particular pair of neurons, A and B. And so there is a change in what presumably represents something like the strength of the connection that's individual to different animals, but the overall pattern is less reliable. And in particular, if there's only one synapse between 2 neurons, that's only about 70% reliable. Whereas if there's more than one synapse, it's greater than 90% reliable across the data set. So you can sort of get a sense that strong connections are very reliable. Very weak connections may be variable between individuals, and people are just starting to put together the thinking about how that might happen. Oh, and then there's also changes in the different sex in the male.
Yeah, right. Yeah. So thank you very much. That's very nice to have that.
Great.
There's one question in the chat, and it's a very broad question, so I don't know if you want to delve in it now or leave it for later, but Yudi is asking, despite its simplicity relative to the human brain, people think we are still far from understanding the C. elegans nervous system. What does understanding mean to you, and how do you define success? So
that's a great question, and I will I'll try to use that question as a frame for the different topics that I talk about in the lecture. So what would it look like to really understand this process is a great question. So yeah, so I would say that to really understand a process means to understand the level at which you need to explain it. Is this a question that is answered at the level of a molecule, a cell, a circuit, a genetic network, or so forth? That's the first part. And then second, what are the underlying principles and sort of theoretical concepts that underlie that explain the outcome? So you don't have to so you need to have a quantitative and not just a qualitative understanding of what that is, in which you can describe, not in every detail, but rather in, quantitative, but not necessarily absolutely precise terms, what is occurring at a given stage of the analysis. So for example, I think the level of explanation we have about the sensory neurons is pretty good, and I'll show you a little bit more about that. It's like the sensory neuron detects this odor, it senses these genes. I'll show you a little bit about its dynamics. I think that's pretty much okay. There's still some more to do, but it's under control, whereas the level at which we understand those integrating neurons is still very primitive. And I would say we're sort of partway through a trajectory there. Yeah. Good question. And thanks for asking, and thanks for kind of pointing out to me what you think good questions are. So here is the next generation. The next generation is not just letting a worm move around freely on a plate, but rather delivering precise stimuli, in this case through microfluidic devices, which are the size of a microscope slide, but allow us to make really well-defined either temporal or physical environments of odor where we know exactly what the concentration is at any point. We can give it in a time pulse, we can give it in complex time pulses, we can give it in a gradient or in stripes, and we can put many different worms inside this device, flowing the odor past them. And we can measure their responses because, as shown here, we can express genetically encoded calcium indicators in small subsets of neurons. Here, the individual neuron pointed out with a green arrow in each worm. And then we can define the properties of those neurons by looking at their activity. And what was absolutely missing from the wiring diagram for many years was an understanding of what the activity of those neurons was as opposed to what their anatomy was. And that was very much driven by these genetically encoded indicators and by additional experiments using electrophysiology that I'll tell you about. So what this allows you to do is to invest neuronal dynamics across the different cell types, understanding what the sensory input is and what the transformation is that's occurring at each step in this process. And I think that's a key element of this question of what does it mean to understand the nervous system, is to understand what these dynamics are and how this is transformed in time. How are different odors recognized instantaneously, and how does that convert into the 15-minute track that I showed you earlier of tracking over a long distance to the odor. And I've simplified here the wiring diagram to a simpler set of cells. What are you supposed to be seeing here? You're supposed to be seeing that it starts with sensory neurons at the top, and at the bottom, we can measure parts of these motor systems that mediate particular outputs. So for example, of the different behavioral outputs, there's forward movement, reversal movements, there's different kinds of turns, and we know what the systems are that act as sort of the command systems for those different outputs. And then in between, there's this heavily interconnected system that gets you from the first point to the second point. And one of the things I want you to see is just the density of the connections among these different neurons. The second thing I want to just point out is that there are things like chemical synapses. Those are the arrows. They can be excitatory or inhibitory, as well as electrical synapses. Those are these resistance signs. So those are gap junctions. And I'll mention that these one of the things we're uncovering is that these can have very different functions within the same little subcircuits. And I'll give you two examples of that. So let's start with electrophysiology. Chung Liu, a really talented research specialist in my group, has been studying the electrophysiology of C. elegans neurons. This is technologically very challenging, but he has set for himself as a task the idea of recording from all of the different types of neurons in the C. elegans nervous system to generate a first level of understanding of what their dynamics is in response to different changes. And so here he does a series of current clamp experiments, measures the voltage output at different current inputs. And what you're supposed to be seeing here is that the responses of these neurons are quite different from one another. So here people used to say that C. elegans that nematode neurons in general were graded. And here at the very left, RM is an example of a neuron that is graded. You give it a stronger input, it gives you a stronger response. You give it a hyperpolarizing input, it gives you a hyperpolarizing response. That too is graded. So it can occupy these, say, 10 different states along a continuum from highly hyperpolarized to highly depolarized. Now, here are some other examples. On the upper right here is a neuron called AFD. This neuron is completely bistable. It has a low state and a high state, and above a certain input threshold, it jumps from one to the other, and below it, it jumps back down again. It's a very clear division of information into two states that this neuron is doing. So that's quite distinct. And then there's a neuron like AIY there in the middle that's kind of got mixed properties, graded for part of its response and then compressed for another part. So it seems like each neuron has not just an anatomical position and a set of connections and a set of genes expressed, but actually a set of temporal properties that it's using to filter the information that you put into it. And one of the things that Chung discovered early on in this catalog was something that we didn't expect at all, that we were very excited about, which is the neuron down there at the bottom called AWA. And it has these sorts of bistable properties, sort of a low state and a high state, but superimposed on the high state, he saw something that we didn't expect to see. And that is what is unmistakably in its shape an action potential, a climb to a threshold, a very sharp peak, a rapid recovery and a hyperpolarization, and then a return to baseline. This is what an action potential looks like. And the reason this was exciting was that it was believed that C. elegans didn't have action potentials. It was believed that nematodes in general didn't have action potentials. And here, in showing that these neurons have very rich dynamic properties, we show not only that they have them, but they show some that they weren't expected to have. And Chung did quite a lot of work to characterize them. It turns out that they are generated by a set of molecules that we can understand in the sense of action potentials. For example, action potentials require a voltage activated channel that opens at a sharp threshold. That's a particular calcium channel in this case. They require a voltage activated channel that opens to repolarize the neuron. Chung was able to define that as a potassium channel called Shaker. And just by putting this together, again, here it gives you an idea of what we're trying to solve. We say this neuron has this property. We can ascribe that property to particular neurons. We can explain it with those neurons. We can explain why people thought worms weren't ever going to have any action potentials, which was that they didn't have the right kind of sodium channel, but they do have a calcium channel that can take that role. So we see it here. And then we can ultimately ascribe this upward to the behavior that they regulate. In the case of the AWA neuron, this is an olfactory neuron. It detects odors, and these action potentials fire every time the odor concentration increases, but then they quickly settle back down again. And so what this neuron is doing is looking for an increase in odor and sending a strong signal for when the odor is increasing, that thereby drives the behavior that drives the animal toward higher and higher levels of odor and allows it to climb up over a 100-fold range of odor concentrations. So lots of other neurons spike too, it turns out. Of course, most neurons in the mammalian brain are known for spiking, although others do not. And I just want to point out that spiking neurons also have really different properties from each other. This is absolutely a feature of neurons in different brains, that there are different patterns of spikes, whether those are bursting patterns or regularly timed patterns, that there are delayed spiking neurons that take quite a while to get started and others that start very quickly. There's a lot of dynamics in the mammalian system that suggests that these properties are going to be allied with particular functions. And putting those two things is a problem that I think we can solve in C. elegans at multiple levels. And just to mention that here in particular, Chung has continued in his analysis. He's recently defined 2 additional neurons that have action potentials. These action potentials are very different in shape from those in AWA. They're different in dynamics and their durations. And he's now mapping those to the behaviors that they control, which are behaviors involved in controlling the enteric nervous system and digestion. So we want to move downward to the molecules and then upward to the behavior through the properties of neurons. So this is giving, I've now told you something about sensory neurons. So we have these kinds of properties mapped. We know some things about the durations over which they interpret behavior, including things like rapid adaptation or slow adaptation in different ways. So now let's try to get into the more difficult part of this. Let's try to ask what a sensory neuron transmits to the downstream neurons. So here's that neuron that fires spikes, and here are a set of its different targets. And I'm just going to, as you can see, it has multiple targets, but I'm just going to pick out one that's the source of really quite strong connections. There are electrical synapses as well as chemical synapses that link these 2 neurons together in what looks like a tight coupling here. But this neuron also receives input from other sensory neurons, and it sends output to a variety of different cell types. So how do we study what it transmits? We want even better control. Again, part of my theme here is controlling the sensory stimulus to understand the properties of the neurons, to separate out the timescale of what the external world is providing from the timescale of what the nervous system does with that information. And in this particular case, May Dovich-Chevitz, when she was a graduate student in the lab, decided she really wanted to refine this. She wanted to refine it so much that she didn't want to be just dealing with the different responses to the odor that arise from the chemical transmission in the cell and the various delays that in the chemical transduction in the cell and the various delays that imposes. She wanted to control this cell completely using an artificial system, using optogenetics or light. So what you're looking at here is an illustration of an experiment where she either stimulated that cell in a controlled environment in that microfluidic chamber with diacetyl, or she stimulated the same cell, sometimes in the same animals, by genetically expressing an activating channel, activating that neuron to lead to its strong depolarization. And then here she records 100 different animals and looks at what happens to the calcium response in those animals. Actually, I think it's about 60 animals here and about 200 animals here. And what you can see is that in response to odor, there's this increase in yellow color, which indicates a depolarization of these neurons, increase in activity to odor. And then here's a response to the optogenetic stimulus, and that too gives rise to an increase in activity coordinated with the stimulus onset. So she can activate the sensory neuron either way. And now using that, she now turns to the downstream neuron here to look at its pattern of activation, and she gets a surprise. And the surprise is that here on the left is more or less what we expected. We saw this sensory neuron coupling quite robustly to the downstream neuron. 95% of the time when you added the odor, the downstream neuron responded. The lag was a second or so, pretty quick, a little slower than you might think, which is interesting, but is so different when she stimulated it with optogenetics. And instead, now only about 50% of the downstream neurons responded at all. When they did respond, they responded with a lag of about 7 seconds on average, which is a really long time and cannot have to do with this optogenetic stimulus, which is basically saturated instantaneously. So this is proposing a puzzle to us. It's saying the odor is giving a nice, reliable transmission of information from sensory neurons into the circuit, but this optogenetic stimulus of equal strength is not. And May looked at it. So what determines whether a sensory neuron versus an inner neuron responds? And the answer is it's the interaction with other neurons. In this particular case, other sensory neurons that I mentioned to you converge on the same inner neuron. So what am I showing? On the right here, I'm showing you that while diacetyl activates AWA, which it does quite strongly, it also weakly inhibits several other sensory neurons. And we didn't pay a lot of attention to that because it wasn't required for the chemotaxis behavior, and it was a bunch of them, and it was kind of weak, and it was distributed. And amusingly, it's been a property of the data since the 90s, like that we knew that this was true, but we just didn't pay any attention because it wasn't the big strong effect. But now that we're looking at these intermediate neurons, we realize that that's actually really important, because these neurons here are inhibited by odor, and they inhibit this downstream neuron, actually through chemical synapses. And the downstream neuron, in order to be activated, needs both this key central controlling AWA neuron to be active, and it needs these other neurons to be off. And so it's a combination, it's an integrating function that this neuron has that ties together the way that sensory information is being propagated against a field of sensory neurons to develop a fairly strict requirement. And on the left is just one experiment that showed that when May shut off the synapses from these neurons, now all of a sudden she could get a completely reliable transmission of information from the sensory neuron to the inner neuron. So we can use, again, this kind of information to get a richer sense of the wiring diagram than we got from our initial AWA senses diacetyl picture. We realized that multiple neurons sense it, that what the inner neurons are looking for is integrated sensory information, and that they are tuned to that information, and they will only respond reliably when the right combination of neurons are activated or inhibited in this case. And we can also put that together to say, well, one of the reasons this is the strongest input is that it fires that action potential that is strongly driven to the inner neuron. But that alone is not enough. The inner neuron also has to shut off a lot of inhibitory inputs that come from other sensory neurons that participate in its function, which is intrinsically bistable. So it needs both of these things to be in the right state to drive it to the high activity state. And again, this nice intrinsic bistability, now we have a way of thinking about what it's doing functionally. And the bistability was, again, discovered by Chung Liu, this amazing electrophysiologist in the lab. So this is a level of description. There's still more things to know about this one, but I think this one's actually pretty good. The next inner neuron I'm going to tell you about, I think we are only at the very beginning of understanding what it's trying to do. And to tell you about that, I'm going to tell you that we can really monitor neuron and behavioral responses to odors in these microfluidic chambers at the level of individual animals stimulated many, many times with the same odor, and then look at the calcium responses in a given neuron when the odor is delivered. So now I'm switching to a different sensory neuron. This neuron is inhibited by odor and activated when odor is removed. And what that little movie of a worm showed you was an animal moving freely, experiencing odor, continuing to move forward freely. And then when the odor was removed, it generated a reversal. This neuron is detecting attractive odors, and when that odor disappears, the neuron becomes active and the worm reorients or reverses to try to get back into the odor. So it's sort of the opposite computation from what the neuron that's firing action potentials that's driving itself forward into the odor is doing. This neuron, through its patterns of activity, is trying to avoid odor decreases, where AWA was trying to follow odor increases. And we can track that and look at the same time here at the calcium response in that neuron and see that it is matched in this case to exactly when that behavior arises. So let's take that and look at those neural and behavioral responses. And this, kind of very impressionistic checkerboard here shows you about 400 individual times that an odor was delivered to a freely moving animal and what it did. And what you can see is that sometimes it kept moving forward. It didn't respond at all when the odor was removed. Sometimes it generated a reversal, which is something you saw. And sometimes it generated an elaborate reversal turn sequence that we call a pirouette after the move in ballet, where you turn multiple times. And these things, individual animals would show all three of them. Any individual trial could show any of the three. It's not getting stronger or weaker over time. There's some kind of what appears to be a random or unreliable pattern. So same motif as AIA, pattern is unreliable, but I'm going to tell you that has a different answer to why it's unreliable. So where's the variability? Where does variability arise? So here's the neuron that's inhibited by odor up here. And now I'm going to go through the targets, because I told you this neuron is very good at driving a reversal when the odor is removed. So let's look at the neuron that controls reversals. That's this guy down here at the bottom. And let's chart the best path through the wiring diagram to that reversal neuron. And that goes through these two intermediate neurons, AIB and RAM, although there are other paths as well. So let's think about these, particularly about these integrating neurons, and let's follow information from the sensory neuron to each of these integrating neurons out through the bottom here. And now what are we doing? We're using genetically encoded calcium indicators again. We're using well-defined odor stimuli, adding and removing them, and then we're looking at how each neuron responds. First answer.
There's a question on the board, chat room.
Sure.
Yeah, Yudi, why don't you ask the question directly?
Yeah, so could you, like, it's a question about the behavior, the video that you showed like 2 or 3 slides before. Like, it seems from the behavior that, like, you're saying it is like kind of avoiding the direction of the decrease in odor, but the behavior seems to like after the reversal, the worm seems to move in the original direction.
Mm-hmm.
So right.
Yeah, in short reversals, which is what that was, the animal will tend to continue in a direction just at a slight angle from what it was doing. In pirouettes, it basically will come out of the turn almost at a random direction compared to the start, although actually biased away from it. So it can use reversals. So a short reversal like the one you saw will often be followed by about a 30 to 60 degree change in the angle from the starting point. So they'll come out not quite at the same place that they did, but close. And a pirouette could be 180 degrees or 120 degrees or more broadly. So it kind of can explore that whole, it can explore the whole dimension.
I also had a quick question about the choice of behaviors. Is this choice of pirouette, reverse, and forward kind of an arbitrary choice? Because I guess there are many ways you could categorize behavior, or there's.
Yeah, these were categorized. They're categorized originally based on statistical properties. And increasingly now based on neuronal properties. So for example, pirouettes, which involve these sharp turns, require a set of neurons that are not required for reversals per se. So, and that was something, it's called an omega turn actually, and it's shown in one of those diagrams there. And so there's a different neural basis for some of that. There's some different sensory basis. Weak stimuli will generate a reversal without a turn. Stronger stimuli will generate a reversal with a turn. And then finally, the timing of some of these different things, Sean Lockery looked at very early on and showed that just sort of like the clustering of particular turns and the timing between them, the length of a forward state between those different turns was also characteristic. So you use sort of the statistics of the behavior, the properties of the nervous system, and the response to sensory inputs to categorize these. There are a number of different things an animal can be doing when it's pirouetting and reversals. I don't want to say it's just, no, there are these 2 categories, but these are pretty clearly 2 categories that might in the end turn out to be, 2 categories and 6 subcategories or something like that. Okay, it's a reasonably good description under these circumstances in this device, in the absence of food. And I'll show you that some of these things actually look quite different. A little later, actually, I'll show you. Yeah. Okay, so what is this slide? This slide is just, again, emphasizing that we have lots of data and we can look at all those single trials. And instead of categorizing them by worm, we can categorize them by what the behavioral response was. And then we can ask, how did the sensory neuron respond? And the answer is the sensory neuron responds equally well in every worm, really quite reliably, characteristic dynamics, regardless of what the worm did. So the worm knows that that odor was there because the worm can tell us that odor was removed through this genetically encoded calcium indicator, but it does not always act on that. So the unreliability is downstream of detecting the odor. That's not the problem. Did you want to have a follow-up question? Okay.
Yeah, so my follow-up question is, if the worm's objective is really to avoid the decrease in odor, right, by doing this reversal or some omega turn, right? So why sometimes we see, like, after the reversal, it still resume in the original direction? So this seems like a suboptimal strategy.
This is a strategy that's actually been really well characterized in in bacterial chemotaxis originally by Howard Berg and colleagues, really one of the first computational biologists and quantitative biologists, a really remarkable person. It's called a biased random walk, and it turns out to be a suboptimal strategy if you are in the perfect environment and can move perfectly toward a target, as you say. But it turns out to be an optimal strategy for animals that are not actually completely in control of their own movement through the environment. So or cells. So if you are in turbulent environments, or in particular, if you operate at a high Reynolds number, you actually can't generate completely reliable responses to the environment around you. And so it is a better response to generate somewhat variable responses, because that will, if you have a very strict strategy and it doesn't work, then you're doomed. Whereas if your environment is turbulent, or whether you're so small that you're actually not able to generate exactly the movements that you want, because the effective viscosity of your environment is much higher, which is this Reynolds number issue, which actually affects both worms and bacteria, worms to a lesser extent, you're better off generating a trajectory that has some degree of stochastic response, because it lets you explore the environment. So it's called diffusion with drift, actually. So you don't, you bias the responses, but you don't create them completely deterministically. So yeah, so again, you can kind of work these things out, and Sean worked it out, and that relates to that first trace I showed you of a worm moving to the gradient. It was showing, it was moving up the gradient, but it was reorienting. It was by no means a straight line. But if you think, so it's sort of, it's definitely doing what appears to be suboptimal if you know where you want to be. But in the environments that it really finds itself in, it has to be able to generate a little more randomness. Actually, just to kind of, it's important, machine learning actually has some of the same features, where you deliberately, you don't want to get trapped in local minima. You don't want to get trapped in a place where in every direction it seems like you're going in the wrong direction. And so you actually generate a certain amount of randomness to jump you out of that, to look for a true global minimum.
Yeah, I see. Thanks very much.
Yeah. No, thanks for asking. It's good to know what people find interesting. Okay, sensory neuron is reliable. Sensory output, we can monitor the release of neurotransmitter at a single neuron level in vivo in response to odor stimuli. That was the thesis work of Donovan Ventimiglia in the lab, and that too happened every time the worm detected odor and odor was removed. It gave a very sharp response. So it's not the synapse that's unreliable. What's unreliable is the interneurons. So if you look at the very first interneuron, you see that sometimes when you add an odor, it becomes inactive, but sometimes when you add an odor, it just keeps responding. And when you remove an odor, sometimes it becomes active, but sometimes it does not. And so there's a lot of variability in this response crossing the synapse. That should remind you a little bit of what you saw with AIA, but where this one's really different is that this work of Andrew Gordis, when he was in the lab, looked at several other interneurons at the same time, some of these downstream neurons. And those downstream interneurons were active at the same time as AIB, and they would all turn on and off together. And so that there was some sort of a coordinated transition, a coupled transition between a series of neurons that was happening, not just a response of a single neuron to a single odor. And this basic observation has been extended greatly by progress in the field. I'm showing a slide from Manuel Zimmer's lab here in Vienna, but there's several other labs that have been doing this as well, notably Aravi Samuel and Andy Leifer, where people are recording from large numbers of neurons in the worm brain, whole brain calcium imaging. And what they're seeing here in a trace like this from Manuel's lab is that many neurons have participate in coordinated and collective activity states. So actually at the top row of this this table shown in dark red are some of the neurons that Andrew studied. And you can see that those neurons over a long time scale of like 1,000 seconds actually of spontaneous activity were always active at something like the same time. And then if you look at the second group of neurons shown in light red, you see that those neurons are active at a different time. They're anti-correlated with the first set, but they're all correlated with each other. And then there are other neurons that have more complex patterns in this third set. And it turns out that if we go back to our historical laser ablations, these neurons in the top set are neurons that are involved in reversals. These neurons in the middle set are neurons that are involved in forward movement. And these neurons in this third set here are involved in turns, in sharp turns and omega turns. So we can actually divide the information up so that groups of neurons are simultaneously apparently active during different behaviors. Now, they're not always exactly the same. There's clearly some ambiguousness and some ambivalence in this, but the driving activity state of these neurons is collective. And this idea of collective activity and of neural states is emerging in many, many different systems. It's been one of the big surprises of mammalian neuroscience that activity states are propagated through the whole brain, particularly locomotion states. So if you look in V1, which is the primary visual cortex of a mouse, the very first relay to get visual information, that V1 neurons are as dynamically regulated by locomotion and motor activity as they are by visual stimuli. So there's a recruitment across the brain of neurons into coordinated activity states. I think this is fascinating. I think it must be relevant to sort of matching sensory processing to behavior and really creating a unified response. I think this is a great idea of something. This is the example of something I think we do not understand this at all, and it is going to be profound. And I'm really excited about that. I'm excited about the idea that worms will make some contributions to it too. So more or less what I told you here, here's a summary of one idea, is that odors are trying to drive a behavior. The sensory neurons are giving input that can drive a behavior, but they're competing with a motor circuit that might be at different times inclined to drive a different behavior. And that there's information flowing through the circuit from the sensory neurons down to AIB here, but also flowing upward from the command circuit and kind of keeping the whole system in some sort of a coherent state to give rise to these switching between collective states. And I just want to say one more thing about this switching between states, which I think is just really at the heart of an incredibly interesting problem here. I want to highlight a neuron that we've been studying in that switching process, this neuron down here in green. And this neuron has sort of an interesting role. It's not, that it is required to keep odors from driving the circuit. That we can silence this neuron using a particular genetic reagent. It's called HisCl. And when we silence that neuron, now, instead of odor sometimes driving the AIB neuron, odor drives that AIB neuron every time. So now odor gets to drive the circuit. And not only that, we're looking here at the backward command neuron, ADA. Odor doesn't drive that efficiently, sort of at baseline, just a fraction of the animals respond. But if we silence RAM, AVA now responds quite reliably, going from on to off with odor. So we can see that a neuron is actually involved in this switching process between the sensory and the motor output. And from recent work of Elise Sordillo, a graduate student in the lab, she's even been able to describe how it is carrying out that function in the motor circuit. And this, again, I'm not going to hit it too hard, but I'm going to mention that this is another case where chemical synapses and electrical synapses are doing different things. In this case, the chemical synapses from this neuron are stabilizing reversals, and the electrical synapses are destabilizing reversals and stabilizing forward runs. And without going into the details here, I think this is the kind of information that are the very kind of first little clues about what these stable collective states are, that this is helping us understand something about how the worm switches between them.
Cori, excuse me, if I may ask a question. Here in these switches that you're describing, so I have two questions, I guess. One is, are these happening in what you call spontaneous activity, or you're stimulating the worm somehow? And secondly, what is it exactly that you call spontaneous activity? So it's a worm which is placed in gel without any explicit stimulation, but how can I characterize this spontaneous activity? Is it kind of a persistent state, or do I have neurons that are generating some regular input? Or can you say a bit more, what is this spontaneous state?
Yeah, so it's a very good question. It actually points to a weakness in this body of experiments that needs to get better than it is now. So there's two forms of spontaneous behaviors, which we monitor in freely moving animals in well-controlled environments. And we can kind of monitor things like how long are forward runs, how often are they interrupted by pauses, by reversals, by turns, and so forth. So we can generate large-scale statistics of the behavioral level. But when we look at neural activity, there's a much smaller, we're just getting at the point, and Lewis is one of the people who's really at the frontiers here, of being able to look with a lot of precision at a lot of neurons and their activity patterns during spontaneous behavior. So most of the experiments that people have done, and all pretty much of the experiments that people have done looking at many neurons for these collective states, they've done in trapped animals within a microfluidic device. And those trapped animals have patterns of activity that correspond to neurons being active at times that if the animal was moving freely, it would be reversing, or it would be moving forward, or it would be turning. And so this is what in the neuroscience field people call fictive locomotion. They like convince themselves that the animal thinks it's moving, but it's quite clear that the dynamics are somewhat different. So for example, worms in a chip spend a lot of time in the reversal state compared to worms freely moving. And there's really clear similarities in Drosophila. You can show that a Drosophila walking on a ball has one pattern of neural activity. And if you take the ball away, it starts doing what's called flailing, and it's a very different pattern of activity. So we're looking at spontaneous activity states. They're related to forward, reversal, and turn states in the patterns of neurons that generate them and the patterns of synchrony that generate them. But the deeper experiments will require animals that are either moving freely or at least showing activity states that are closer to those that they would be generating moving freely. So step one. Mm-hmm. Yeah, very good question.
Thank you. I thought Claudius had a question.
Yeah, I saw a hand up. Yeah.
Yeah, thanks. I have a very basic question. How much plasticity is in this in this behavioral control? So can they learn things and can you change the statistics of reaction to odor upon exposing them to some nice odor or bad odor? I don't know. Or is this something that is all hard-coded, these interactions of inhibition and...
Yeah, I'm not going to talk about learning, but C. elegans actually does learn. It has a number of, so it can learn to, it can strengthen the responses of odors that are paired with food. It can weaken the responses of odors that are paired with food deprivation. It has a very rich form of learning in response to pathogenic bacteria, which are basically a natural predator, where it learns to avoid the odors of bacteria that made it sick. And not only can it learn to do that, which those are memories it'll retain for 24 hours or more, it can actually pass that information on to its progeny. And there's a set of papers from a woman named Colleen Murphy at Princeton that characterize that. It uses like small RNA mechanisms to actually create a stable molecular representation, whereas just the sort of odor plus food, odor without food is more classical, short-term remodeling of synaptic circuits, but not understood not understood completely. So there's a body of work there. It is definitely a next frontier. It's definitely something these animals can do. Yeah, and there's other forms of plasticity too. Mm-hmm. Yeah. So I want to touch on one more topic, and that is the role of modulation and internal state in modifying behavior. And I want to say this, I think, especially because I am a neuroscientist, and if you think about what a psychologist calls a behavior, if you think about psychiatric disease, if you think about drugs and abuse, those are not things that are involved in the rapid processing of sensory information. Those are involved in these motivational and emotional states. And they just completely remodel behavior. And they're controlled by a special part of the nervous system, even in humans, these deep brainstem nuclei. And they're even controlled by a different set of molecules than control the kind of fast behaviors I showed you earlier. So fast behaviors are largely about ion channels. But these slower behaviors, arousal states and so forth, are controlled by small molecules, modulators like dopamine and serotonin, or neuropeptides, of which there are like 1,000 neuropeptides in 100 neuropeptides in the worm genome or in the mammalian genome. And all of these signal using biochemistry and not electrophysiology to a first approach. They signal through G protein coupled receptors. And the timescale of biochemistry is a little slower than the timescale of electrophysiology. So they're modifying the functions of neurons on somewhat longer timescales to create these kinds of coherent states that might last for seconds or minutes or hours. And one of the things that we learned about this a while ago, really through work of Hien Zhang, when she was a student in my lab in collaboration with Kyu-Hyung Kim and Piali-Sen Gupta at Brandeis, is that these modulators are reshaping the wiring diagram. They are changing the way information flows between different systems. And Hien and Kyu-Hyung showed this with response to pheromones, because pheromones, unlike diacetyl, are not always attractive. Sometimes animals want to aggregate, other times they want to avoid each other. It's regulated by stress, it's regulated by sex, it's regulated by feeding state. It's a much more plastic behavior to whether you respond to other animals. And they showed that there was always the same olfactory neuron that sensed a group of pheromones, but the animals could switch between being attracted to pheromones on the right, or being repelled, and form these aggregates, or repelled from pheromones on the left, under the control of a neuropeptide receptor and neuropeptides. And that what these molecules, which are associated with high arousal states and low arousal states, are doing is basically turning off one set of synapses in the wiring diagram and turning on, allowing a different set of synapses in the wiring diagram to be on. So when the neuropeptide receptor is active, the gap junction circuit is turned off and attraction is not observed, and instead repulsion is observed. When the neuropeptide receptor is inactive, in this case, the gap junction circuit dominates and you see attraction instead of repulsion. So I think, why don't we understand the wiring diagram? That was one of the very first questions I got in this discussion. When will we know what it is? Well, the wiring diagram includes in its coding two completely different behaviors, attraction and repulsion. And that's just there in that static diagram. You can only understand what's going on by understanding the state of this neuropeptide within that system. And so we think this is a really important contributor, and this needs to be understood. And I want to remind you, there's 100 different neuropeptides, and there's like 5 different modulators. So there are a lot of ways to rewire this nervous system in this kind of biochemical way. And so one of the things we really want to understand is how do these neuromodulatory circuits relate to the synaptic circuits? And so we have our wiring diagram, again, our favorite part of the wiring diagram with different kinds of behavioral outputs. And then we can try to ask how modulators affect that. So one of the things that we see is that the modulators tend to modify behaviors for longer periods of time. And they give rise to spontaneous or pattern behaviors, even in uniform environments, that can show us the animal switching between these different kinds of arousal states or motivational states. In this case, there's a state called dwelling in blue. It's a calm state. Animals will stay in one area for 10 or 15 minutes, moving slowly and feeding. And then there's a high arousal state that they will enter into completely spontaneously and move rapidly to a different direction for a minute or two, and then they'll find a new place and they'll just sit there for a while. And this is without any inhomogeneities in the sensory signals. We just see this kind of a difference in this behavioral output. It seems to be internally generated. So using this behavior, which had been described a while ago by Manabe Fujiwara, Steve Flaubel, when he was a postdoc in the lab, discovered that neuromodulators have a really important role in determining these kinds of long-lasting behavioral states. So we think these are worm analogs of like a motivational state. So animals that lack serotonin spend a lot of time in this high activity roaming state, high speed, low turning activity, compared to animals that have serotonin present. So that lets us monitor the function of a modulatory system. And in this case, we know exactly what neurons make serotonin. Some of them are connected to the foraging circuit. Others are quite distant from the foraging circuit, for example, in the feeding circuit. And what Steve did was to knock out the serotonin synthetic enzymes in each of these different neurons and ask, where is serotonin important to generate and regulate these roaming and dwelling behaviors? And what he found, to our surprise, is that it wasn't the serotonin neurons that were directly connected that were important. It was neurons acting at a distance, what we think of as a wireless or distributed circuit, where actually neurons that are involved in things like feeding and egg laying are determining how these foraging behaviors are going to be regulated by acting on receptors of neurons within the foraging circuit. And similarly, there are other kinds of modulators that change the circuit in different ways that have these same properties. The modulators are coming from kind of outside the main circuits, acting at a distance and modifying them. And I just want to remind you of that picture I showed you of the human brain, where the neurons that make things like serotonin and dopamine are deep in the brain, but they're projecting all over the cortex. This really looks like a pattern that reflects these systems. So we can start to look at what these circuits are. We are at our infancy of understanding what these are, but we can say that they don't follow the wiring diagram with wiring. They're orthogonal and distributed. And we believe they select among connections to reroute information flow, to switch animals, for example, from a pheromone attracting to a pheromone repulsing state, or from a high activity state, a high arousal state, to a low arousal state. And in some way, they're organizing higher order coordinated circuit states that are actually affecting things like those patterns of forward reversals that I showed you earlier, the shorter ones, but they're longer lasting. They last over minutes to hours, and they reshape these other kinds of states to give rise to different patterns. How often do you reverse within a dwelling state is much higher than how often you reverse in a roaming state. So the statistics change based on these modulators. And I just want to close on one final point, which is there's no reason to just stop by looking at these things over periods like an hour or so. Shai Stern in the lab, inspired by Steve Flavel's work, decided to track individual animals for all of development in well-controlled environments. And so here is an example of about a 60-hour experiment looking at individual animals, each per row, at different times of development, and showing that there are actually quite reliable behavior patterns across development that are shared among different animals. All the animals are active early in their third larval stage, but then show very complex patterns that we can analyze further. So another aspect of behavior that we can study over still longer timescales. So I'm going to wrap up now for more real questions and discussions. So what I just told you is that there's a foraging circuit, and we've been doing all the studying of it, but it's not just one circuit, it's many circuits. That neuromodulators are coming in here and changing the activity of the neurons in the foraging circuit to change what output you can get, even to different sensory inputs or to spontaneous activity. And that the neuromodulators are reshaping this over the time scale of minutes, but also over the time scale of development. Second, I told you about neural dynamics and sensory integration, that neurons with different properties, like graded properties or spiking properties, neurons that are activated by odor versus inhibited by odor, that this information, and these neural dynamics are being interpreted by downstream neurons that act as sensory integrators to put this information together, so it's not so distributed and dispersed. And finally, this kind of incredibly interesting property of collective dynamics, where sensory and motor circuits are active at different times and are sort of, the motor circuits are actually continuing to propagate information. That is, it's more as though the system is a motor system where sensory input is modulated rather than sensory input driving the motor action most of the time. And finally, in answer to the earlier question, there are in fact many different forms of learning, and those are being manifested by these same circuits. And we don't fully understand how those circuits are being modified during the processes of, for example, memory formation and memory retrieval, which reside at different locations in these circuits. So this is my conclusion slide. The wiring diagram changes over time. That it can change through development, it can change through modulators, it can change through circuit states, and that imposing that fourth dimension of processing on this anatomical dimension is going to be a really important and interesting part of understanding the behavior and of understanding what it means, how that information is flowing at some times and not others. And I'll just show you a picture of some people in the lab who are from a recent lab picture. I want to highlight here Chung Liu, the electrophysiologist, who really is grounding this in a much firmer set of data that help us to understand it. But then I also, I just want to close by saying that, that how much you don't do science alone, and how much the science you can do is determined, for example, by the technology that's available and the collaborators that you have. And so, there were many, many different techniques that were developed, in some case over dozens of years, or data sets like the wiring diagram in the Genome Project that just have informed all of the work that I told you about. And it's really, in the C. elegans field from the very beginning, we really understood the importance of generating large data sets, really well annotated data sets, and sharing them with each other, and using technologies and developing them. And I do think the next level of large technologies that's really important for neuroscience in general, but for biology in general, is going to be theoretical and computational approaches. That as we've gotten better at generating high quality data, and that this is going to be the level at which we can understand the rules that are governing the processing of information and the way it moves through these systems. And I guess I want to say, just to sort of end, that I really believe in this notion that these kinds of approaches are important. I also recognize, many of these methods came from chemistry and physics into biology. It's so important for people to be able to collaborate across different fields, because you can't be an expert in everything. And so I think it's going to be incredibly important for experimental biologists to collaborate with computational biologists, with physicists to develop new technologies, with chemists to develop new biological probes. And I think this is really the future of our field, is bringing people together across these disciplines for deeper understanding. And I'm doing that, in my own part of the career. I still have a lab at the Rockefeller University, but I am also spending a lot of my time now working for a new philanthropy founded by Mark Zuckerberg of Facebook and his wife Priscilla Chan, that's doing biomedical research. They're going to be giving away the majority of their wealth in their lifetime for this philanthropy. And when I asked them what they wanted to do, they said they wanted to support science and technology that will make it possible to cure, prevent, or manage all disease by the end of the century, which is 80 years away. So we have a long time. And I don't think it's impossible, but I think for that to happen, science is going to have to move a lot faster, and we need to accelerate science. And I believe, based on my own experience and based on my knowledge of the field, the way to accelerate science is to develop new tools and technologies and to support collaborative research. And we're very interested in supporting computational and theoretical approaches, but tool development in general, we really feel like this is where the progress is going to come from, and that's where our focus is in the first 10 years. And so with that, I will close. I'm delighted to continue the conversation and hear what you have to say.
Well, thank you very, very much, Cori. There were many very thought-provoking parts in your lecture. Thank you very much. We will continue with I think your last comments were nice transition to the discussion we'll have in a moment about more general issues of life and science. But before that, maybe there are some questions, some scientific questions on the talk. Okay, well, while people prepare some questions they may have, I have a kind of fairly general question, which is related to Claudius's question, but maybe a bit simpler, is that you described spontaneous activity, you described kind of responses to stimuli, you described kind of motor behavior. I was wondering if it's possible to do experiments, or if you've thought about some experiments that would also involve the study of the encoding of meta quantities. So for example, the probability with which a stimulus may occur, or some expectation. So instead of presenting the animal with say digitized stimuli, the odor is there or not there, to present an odor with some probability and see whether the animal is encoding both the presence of the odor and the expectation of that presence.
That's a great idea. I haven't thought about doing that, but that's a great idea. Yeah, I do. I actually think, and again, I think that the nervous system works on statistics. That the statistics of the world and that those are built in, for example, to the genome and the way that it works, but that they are also built into the way that it functions on a day-to-day level. So I think thinking about that kind of a problem more carefully. So, statistics. For the past billion years, day length has been 24 hours, right? And so your genome just encodes those statistics right into the genome in the form of circadian rhythm. There's going to be times to be active, times to be inactive, times to eat, whatever. And then there are other things that are completely unpredictable. And those things are going to be processed by the brain. But most stimuli fall somewhere between those two extremes of something completely unpredictable that's never happened before and something that happens on some regular frequency. And so just sort of understanding how those get encoded. An example, I think, is this transgenerational effect of, when you're an animal like C. elegans, whose life is only 4 days long, if your parents were starved or if your parents were infected by a toxic bacteria, there's a good chance that will still be true in your environment in your generation. And so that you can think that the statistics of the animal's short life, how much its environment changes on what time scale, could actually be affecting it being physiologically somewhat better suited to starvation or being well fed. Oded Rechavi has done some nice work on that. Or even, what its olfactory preferences is, kind of like walking into the environment. But I hadn't thought about it, looking at it more instantaneously, and I will go back and think about that. That's a great thought. Thank you. Yeah,
I think even I never worked on C. elegans chemotaxis, but I worked a bit on E. coli chemotaxis, and there you can see that the strategy they should use depends on whether they have memory or not. And as soon as you think that they have some degree of memory, you could view chemotaxis as a kind of statistical learning also. But I was wondering if there is a broader, a more direct way one could probe statistical learning in C. elegans, because that seemed that it would be, but one would have to find the behavior where statistical learning is important, I guess, and go for. I would love to discuss this more. It's something that interests me a lot. So.
Oh yeah, it'd be fun to it'd be fun to think about that. And I had a thought. Okay, yeah, great idea.
So any other questions?
Maybe I should ask a silly question. So I've been, since the worm has 302 neurons and it can do all these computations, and I have 10 to the 11 neurons, but I don't feel much smarter in some sense. So what seems to be, am I thinking about the problem in the wrong way?
So here is how I think about it, and it's actually quite relevant to what Raza just talked about. Every animal, every organism that is alive today has been evolving on Earth for the same period of time. I mean, one way of thinking about this is that, there have been the same number of worms on the planet for the past 30 million years. And yet every worm can have 300 progeny every 3 days at maximum. And so that basically natural selection means that worms will have gotten really, really, really good at being worms. That the one out of those 300 progeny that solves problems the best is going to be the next progeny to grow on. Okay, so I think that the worms will have a really sophisticated response to their own environment. But relevant to Raba's question about statistics and about yours, it's a question you have to, the difference between a worm and you is that a worm probably can only learn things that a worm has evolved to learn. So it may not, the wiring diagram is, it's got some variability in it. There's a difference in the number of synapses from worm to worm. It probably is changed by learning. But there is a sort of a fixed substrate that information can be processed under, and the worm is never going to learn to speak French or play the piano. And so what our many neurons enable us to do is sort of massively, is sort of, one-shot learning, massively complex learning, storing completely arbitrary sequences and arbitrary information. Things, I think that it's not the ability to learn that's important. It's the breadth of different responses. It's the kind of flexibility of matching these responses that really distinguish the larger nervous systems, I would say.
So just, I don't want to take too much time, but how about this experiment? Could you do an experiment where the worm has to decide to take or not take some nutrient, for example, or go for an odor or not go, and maybe not go because either there's a competing odor or maybe there are adverse conditions like low or high temperature. And then you could change the intensity of the odor, and you could also change the probability with which the odor appears. Or something like this. And then now you have two variables you can change, and you can look at how the behavior changes as a function of these two variables. But then those two variables presumably are encoded very differently in the neurons. I have no idea whether this is any kind of a doable experiment, but is it something? Can you make C. elegans choose between options?
Oh, yes. Yes. In those microfluidic chambers, you can give them competing gradients, for example, so that they can go, up one gradient at the expense of the other. You can create more complex environments, and also those gradients can change. So you can like flip them around and kind of move them within seconds or subseconds, so that the animal is switching. The environment is changing, so that predictions, for example, are changed. That the relationship between a prediction of A goes to B or B goes to A, you can generate those kinds of information. I'll have to think about it, because I do have to think about, what kinds of problems the animal would actually have to solve. But I think it's a solvable problem, and I think it's an interesting question. I think that, and I don't think Lewis's question was silly, actually. I think it is, you do want to think about what these things are determining that you can, what the constraints are, and to kind of formulate formulate increasingly precise questions that derive from general questions.
Any other questions? Yes. Joshi, you have a question, please.
I have a simple technical question here. So maybe Professor, you already mentioned about it, but I missed it. How many neurons we can simultaneously recording at the same in a C. elegans?
At the moment, people are mostly recording, I would say, at the moment when people say they're doing, recording all the neurons, they're really recording about a third of them. That's still pretty good. They're probably really getting good traces out of maybe 100 out of the 300. There are a few complexities there. They're not getting all of the information from all of the neurons. Every experiment is a compromise. So some of the neurons, they didn't have local processing of information, where one part of their axon processes information differently from a different part of their axon. And this is a property that graded neurons show in other systems too. It's a property that what are called starburst amacrine cells show in the human retina, for example, where like an individual process will process one piece of information and another process of the same neuron will process a completely different piece of information. So that you cannot see in these whole worm imaging experiments. It's at the limit of resolution of the light microscopes, and if there's a bunch of things going on at the same time, it's hopeless. So you're capturing a coarser level of information about a lot of neurons in that experiment. And then, then you can look at Chang's experiments. He's capturing extremely refined information about, in some cases, like one neuron a week. So you're making these different, and then the calcium imaging is kind of in between. You can get a lot of neurons, and the information is, you can get some of the information about things like the processes separate from the cell body, and so each experiment has its own strengths and weaknesses. And I'm very excited about the way that the whole worm imaging work is going. It needs to be moved to free free moving animals to do more of that now. But there's still, I do think that it's such an interesting observation to look at these collective states, that it doesn't really matter if you can't see all the neurons in the collective states or all of their processes. At least right now, you kind of say like, I just want to look at the group. Yeah.
So I know in Drosophila larvae, they do in New York University, I guess they have the machine do the 3D, as you said, like a whole like whole body 3D calcium image, like in a vivo state. Is it also possible now with C. elegans?
I wouldn't, I'm not sure is the answer. I don't know. Some of these things, I don't know.
It's just really exciting about this.
Yeah. It's close. Yeah.
Any other questions? So if there are no more questions, Cori and everybody, should we just continue, or do you prefer to take a 5-minute break?
Let's just continue.
Tell me.
Yeah. Or,
yes?
actually,
okay.
Well, you're right. Let's take a 5-minute break. Let's take a 5-minute break and then come back.
Okay, so let's all meet at 12:30 in 7 minutes.
Good.
Perfect.
Great.
Sorry guys for not managing my Wi-Fi and hotspots very well.
No, I know that's totally fine. I just regret that I didn't have something more coherent and thorough to say when I introduced Cori, but
I was going to read her entire Wikipedia entry, so you actually saved us some time.
I see. Okay, I'll come back in 5 minutes.
Yeah, me too. I need to finish my dinner.